We are fortunate to have Dr. Mansoor Khan provide the next lecture. Dr. Khan serves as professor and vice dean of Texas A&M University College of Pharmacy. Prior to joining Texas A&M in 2015, he served as the director of product quality and research and senior biomedical research scientist at CDER at the FDA. While at the FDA, he led the research and review teams to promote manufacturing science of new generic and biosimilar drug products. Dr. Khan received his PhD degree in industrial pharmacy from St. John's University in New York. He has published over 325 peer-reviewed manuscripts in pharmaceutical formulation and manufacturing science. Dr. Khan has served as FDA representative to the World Health Organization, the United States Pharmacopoeia, the European Medicine Agency, DARPA, the National Institutes of Pharmaceutical Technology and Education, and the International Pharmaceutical Federation. We hope that you enjoy today's presentation.
Hi, greetings everybody. It's great to be with you all. My name is Mansoor Khan. I'm a professor and vice dean in Texas A&M University. And in Texas, we greet people by saying howdy. So howdy, everybody. So first of all, I would like to thank the organizers, Dr. Lisa Cardis and Rebecca Wong, for inviting me for this presentation. And let me pull my presentation here one sec. Well, now you should be able to see my screen here, hopefully. So the title of my presentation is Considerations in the Development of Biologics. So I have a background in product formulation and bioavailability, bioequivalence. So I've been a clinical pharmacologist in Food and Drug Administration before. But I think the presentation will focus more on the development and regulatory aspects of the biologics. So as you can see, there are things that we miss as scientists, and I see a lot of discoveries that they miss. They don't see the light at the end of the tunnel, and so hopefully with this presentation you'll focus on some of the things early on, so that your developments and discoveries lead to some meaningful product development. So a lot of it will be FDA, the development process, and then the biologic characterizations, the protein characterization, right? That's what I will deal with. So the learning objectives, actually, instead of saying outlines, because this is a part of the course, it's pretty much the learning objectives of the outline, the broad outlines here. Let's differentiate between the laws, the regulations, and the guidances that will help us look for appropriate information when you need as you are developing protein formulations, right? So let's outline the FDA drug approval process and describe the IND phases and various types of INDs. And we will describe the various types of FDA meetings. And I'll tell you why these things are important. Once we have these backgrounds, then it's easy for us to understand the development of proteins as both a drug substance as well as a drug product. So where does the FDA derive its authority for drug approval and as well as the compliance activities, right? So the Constitution gives we have legislative function in the Constitution, the executive and judicial. So FDA, as you can see, the Department of Health and Human Services comes under the executive functions, and Food and Drug Administration is within that. So Congress, they make laws, right? So once the laws are there, laws are very, very broad, right? So once the laws are there, the FDA, they will interpret the laws and they will have regulations there. And it will come in the next slide. So they have regulations and then they provide guidances to the sponsor. So let's see all of them, just one or two slides as a primer for all of them. And then they provide guidances actually, so that we will look at how to do things, right? That's what the guidances are provided. So let's look at the laws. So the relevant laws, some of the most important ones, the relevant ones, these are legislations passed by the United States Congress, right? Those are the laws. For example, the Food, Drug, and Cosmetic Act of 1930, all the drugs have to be safe. And PDUFA, prescription drug user fee, if you are developing a formulations and then you are paying a fee for it. And so GDUFA, if you are developing a generic drug product, you're paying a fee for it. So these are, so Congress says, well, for example, there's a Biosimilar User Fee Act of 2012. So Congress says, all right, for a biosimilar product, a protein or biosimilar product, charge a fee. But what kind of fee? How do you do that? How much do you charge? And how much? So what kind of applications? And how do you charge? And those kind of things, they come in either regulations or guidances. So Hatch-Waxman Act made generic drugs possible. So these are some of the laws that Congress passed, right? So what does FDA do with those laws? No, so by the way, the proteins and the biologics, they come under the Public Health Service Act of 351. So in this act, the biological product is defined as a virus, a therapeutic serum, a toxin, antitoxin. Vaccines come under this, blood, the blood component or derivative, allergenic product applicable to the prevention, treatment, or cure of a disease or condition of human beings. You see, these are very, very similar to the definition of drug, where there is treatment, cure, mitigation, prevention of the disease, right? So the reason I wanted to mention that was in FDA, there's a center here, the CEDAR, that regulates therapeutic proteins like enzymes and other therapeutic proteins and monoclonal antibodies, right? But the rest of the protein products, if they are used for diseases, rest of the protein products, they go to CBER, right? So Center for Biological Evaluation and Research. So if you're looking for guidances for monoclonal antibodies or therapeutic proteins, you're looking at CEDAR guidances, right? For all other products, you're looking at CBER guidances. Now, regulations, these are rules issued by FDA. These are consistent with the laws, right? And for example, if you are really looking at an IND, investigational new drug, then 21 CFR 312 is where you look at, right? If you're looking at a new drug application, then you're looking at 21 CFR 314. BLAs, as I indicated to you, there's a PHS Act. So if you're doing human studies, IRB studies, then 21 CFR 50, right? So those are regulations that you will look at. Now, if we distill down further, then there are guidances. So, all right, we are saying that in the previous slide, we are saying that for INDs, you look at that, right? But how exactly it needs to be done, what needs to be done, and those things are there in guidances. So, and guidance, by the way, these are informal documents, right? So they are clarifying the requirements. And they are non-binding, but it gives a pretty good information about the development. So for example, if you are developing a cyclosporine ophthalmic product, then there's a guidance for it. The last one here, if you're looking at immunogenicity of therapeutic protein products, now we're talking about protein products, right? So if you want to look at immunogenicity, how do you do that? For what kind of products you do that? With what frequency you do that? Which models you use? And those kind of things, you will look at this guidance here from FDA, right? So now you've seen the laws, the regulations, and the guidances. So the basic principle for the mission, the functioning of FDA is no drug can be marketed in the United States until substantial evidence of its quality, safety, and effectiveness has been provided to FDA satisfaction, right? In quality, we are looking at the characteristics of the drug, including its manufacturing. So if you have protein formulation, right? How you're looking at the characterizations as well as its manufacturing. So they look at the quality data, they look at the safety evaluations, which is the relative risk of harm, and they look at effectiveness. And essentially, they're really looking at the risk to benefit ratio. The benefit should always outweigh the risk. Everything has risk, right? No matter what you take, there's a risk, right? Sometimes people say that, hey, this problem has this adverse event, or this problem, this product has that problem or this problem. Yeah, problems might be there, or even the adverse events might be there, but the overall benefits far outweigh the risk. Then only the product gets approved. Right? Now, we are looking in FDA really depending on the nature of the product you are developing. It has different centers, right? For example, if you have food or nutritional supplements or food things, you're looking at CIFSAN. And if you have a developing product for veterinary medicine, then you have CVM, right? If you're developing a device containing some proteins, then you're looking at CDRH center guidances, right? And if you are developing drugs or therapeutics with proteins, then you're looking at CDER guidances. And if you're looking at some proteins, for example, if looking at vaccines or cell therapy products or gene therapy products or blood-related products, then you're looking at CBER guidances, right? So same thing, you have Tobacco Center, you have National Center for Toxicological Research. It's done in Arkansas mostly, and then the compliance activities are regulated by FDA, by the Office of Regulatory Affairs. So FDA looks at the new product reviews. They look at safe manufacturing and handling. They look at provide standards and regulations. They also do research and enforcement function. So I wanted to provide this little bit of background to you so that you will have things into perspectives when you are looking at your discoveries and development of those protein products. So FDA reviews the results of laboratory, animal, and human clinical testing done by companies to determine if the product they want to put on the market is safe and effective, right? So essentially a lot of preclinical work, the discovery work, the development work, the manufacturing work, the scale-up work, the clin pharm work, the clinical pharmacology work, as well as the clinical studies. A lot of things are seen, and only if they're satisfied, the product gets approved. Now, also remember that FDA does not develop drug, right? And and this is not like a national testing laboratory also. So the testing that is done is mostly in support of the review function, or sometimes it's in support of the field functions, right? But it's not like a testing laboratory for anybody else to use. So they do the pre-market review of new human drugs and biologics, complex medical devices, food and color additives, infant formulas, animal drugs. So this is some basic work that FDA do. Now, this is an important slide. It just shows that when a drug is developed, a protein is identified, it shows some function, then these are the studies. For example, the chemistry and manufacturing. So either you are synthesizing a protein or you are deriving it, low molecular weights, or the peptides you're synthesizing, or you are deriving it from some cells, or you are extracting it from some other place, mostly from some organs or different sources. So you have a protein there. So you're looking at the chemistry and the manufacturing. So you all know that a lot of animal studies are done, right? These are short-term animal studies are done. And for chronic, you're doing a long-term animal studies are done. So once these things are done, you see this element here, the formulation development. A lot of people tend to miss this information. I have seen even the proposals that I see in the study section in NIH, a lot of them have a good information about this, a very good information about animal studies, but this information a lot of times is missing. And because of the lack of this information, the products cannot go to the IND. So a lot of products get killed here itself. They don't go to IND. And even if they go to the preliminary, even if the IND is submitted, then later on you'll have a problem during phase 1 or phase 2 or phase 3 studies or in the PK studies. But if you want to cross the barrier from this initial development and discovery, we really need to understand the IND very well. So that way we can focus our studies for some meaningful development, right? So the formulation development is an important aspect here in this one. And same thing, once the IND is done, that's not the finished formulation that's going commercially in the market. Even if the formulation goes in IND, then there'll be a lot of changes in that formulation depending on what stage of the development it is. So eventually, When all the studies are done, that's when the formulation gets logged in. The previous studies, the formulations, you are changing excipients, you are changing process, you're trying different things. So you have a lot of latitude in the development in the initial studies. But as you go down further in different phases, like phase 1 and phase 2 and phase 3, and really at this stage here, at the end of phase 2 or phase 3, the formulations get logged in. After that, you don't want to make changes there in it. It's very, very difficult. So the eventual final clinical studies that are done, it's done with a fixed formulation that doesn't go undergo changes there in it. We will learn more about it. So this stage here in IND, if you want to go, so this is an important thing to know. So if you want to go for IND, once you have your discoveries, what do you actually need to get into humans for testing, right? So really, this thing needs to be understood very well. You see this CMC, chemistry and manufacturing control. This thing needs to be understood very well. You are able to reproduce it. You are able to test it. You are able to consistently obtain data that's consistent, right? So it's not like you're getting some peaks in your mass spec on HPLC and tomorrow you're not getting that peak at all. So what is happening there? Is that because there is no results, or is that because there is no drug in it at all, or something has changed there completely, right? So we need to understand this very well, chemistry. And also what we are looking at is a preclinical data with the animal. Obviously you saw that it is safe in a rodent species, on a non-rodent species. So animal species, you have done some studies there in it. So chemistry and pharm-tox studies, when they're done, then we write a protocol for administering the drug to humans, and that's when you get your you submit the IND to the FDA, because FDA, when they are looking at it, they want to ensure that the medication, if it goes to human, before it goes to human, you've collected sufficient information so that when it goes to humans, then it is safe to them. Its early part is mostly about safety, right? And so that's how the IND is developed. So if we really pay attention to this CMC part, attention to pharm-tox, what I have noticed that a lot of attention is paid to the pharm-tox part, not much of attention is paid to the CMC part, and that's where a lot of the discoveries end up prematurely without even going for the INDs, right? Now the IND, we will see some of them, there are different types of INDs. So either it could be just investigator IND, investigator has done some work and then wants to look at administering humans and test it, that will be an investigator IND. It could be a commercial IND. The intent is to commercialize it, move very fast. For example, you're seeing the vaccines now, right? So you're seeing a lot of vaccines under development, and some of them are in emergency use authorization. They're for commercial purposes, even though they're in IND studies, different phases, but we already know ahead of time that they are for commercial purposes, right? So they are traditional INDs, they are treatment INDs, they are exploratory INDs, they are parallel track INDs, they are emergency use INDs. So there are different types. We will look at some of them that will tell you what kind of studies and that are needed, and attention needs to be paid in certain things. So what do we need to submit in an IND application? There's a form 1571. We fill it out, has table of contents, statement and general plan of what you want to do, investigator's brochure, and the technical qualifications and knowledge of the investigator, sufficient protocols and how you are administering, right, and all that. And then all the CMC data, the pharm-tox data that I indicated to you previously, and previous human experience with that, if it is available, right, provide any previous human experience, or sometimes the experience might be in some other countries, right? You saw some literature, or you saw other people working in this molecule or a related molecule in published journals, or any other regulatory bodies have looked at it. Provide that information if it's available. So this is a minimum expectation, right? And in the farm tox data, what are we providing? Some preclinical studies data, right? To demonstrate safety, to demonstrate efficacy. Again, preclinical is in the animal models, right? So you are doing it in two animal species, rodent species and a non-rodent species. That's what is required in most cases. So demonstrating safety and efficacy, potential toxicities, and optimal dosing. Then identification and quantification. The investigators. So who are the investigators? What are their qualification that needs to be shown? Statements regarding where and how GLP studies were performed, what the studies perform in a GLP environment, a good laboratory practices environment or not. In other words, they're saying that is there a sufficient documentation for repeatability and reproducibility, right? And sometimes the amendments can be made. You submit an IND, then new information comes up, and then you provide amendments to it, right? So we provided safety, pharmacokinetics, and toxicity information, right? And depending on the route of administration, if it's an IV, you provide some information for that. Oral routes, you're providing some information for that you intend to use in your studies in humans, right? So now, IND, there is phase 1, that is very initial investigation, right? After you do your experiments in the preclinical models, you're just deciding to work on humans. So you have applied for an IND and you wait for about 30 days for the IND. And then if you don't get a response for IND in 30 days from FDA, there's no clinical hold. That means you can move forward and start your phase 1 studies. But if FDA has questions, or if they want to put on hold this clinical study, then they will get back to you within 30 days, right? So phase 1 study, it's normally done in 20 to 80 subjects. Most of the time, the study is done in normal volunteers, unless it's problematic, for example, in oncology or places where there could be some serious adverse events. So you don't want to give those kind of drugs to normal volunteers when they don't even have the disease, right? But in all other cases, most other cases, that you use normal volunteers in phase 1 study, right? Usually use anywhere from 20 to 80 subjects. You're looking at some of the ADME studies on that one. Very early safety studies is what you're doing on phase 1 study. But if the phase 1 results come out well, then there are the subjects are safe, and you are able to look at the absorption, distribution, metabolism, and excretion of that one in normal volunteers. If that data looks good, then you go to phase 2, where you look at several hundred subjects, and these are patients with disease under study, right? So you're looking at a cardiovascular condition. So you are looking at patients with that cardiovascular condition. You're working on a diabetic medication. You're looking at subjects who have diabetes. And so usually these are patients. These are several hundred subjects. These are well controlled. So essentially, these are safety studies with some efficacy, safety and efficacy studies. And if the phase 2 studies also go well, then you go to phase 3 studies. Here, that's where you have a lot of trials here, the randomized, well-controlled studies, what you need here, from hundreds to thousands of subjects and multi-center in different places. These are very, very elaborate studies, right? So this is also for patients with disease under study. So there are a lot of efficacy elements here in this one. Of course, safety is there, but you focus a lot on the efficacy of this one. Now, once these things are done, usually NDA is submitted and NDA is approved. If the NDA is approved, sometimes there are commitment for phase 4 studies, post NDA approval studies, right? So sometimes there are some risk evaluation, mitigation that you need to do, or sometimes they need to look at a product after approval to ensure that it is received well by the general population, where it might be going in millions of patients, right? So phase 4 studies are done. So you got a glimpse of how IND studies are done. And another thing I would like to suggest for especially good scientists, the basic scientists, you can have meetings with FDA. They would love to see you. They would love to talk to you. They will give you some advice as to what kind of what kind of studies you need to do. And you can have pre-IND meeting, as you can see here, even before you go to the IND. Sometimes people don't utilize this opportunity and then end up doing studies which are unnecessary, which are unnecessarily elaborate study. And so it's good to have meeting with the FDA and discuss, hey, this is what we have done so far, what else do you need? And before you conduct those studies, it's good to have a meeting. So there are different types of meeting, really. There's called a type A meeting, where if you're already developing a product, there's a problem with that product, you want to meet them. So this is a type A meeting. But most of the development meetings that we're talking about today they are type B meetings. Either you can meet on pre-IND or end of phase 1. Once you complete phase 1, now the question becomes what kind of patients, what's the inclusion, exclusion criteria for that one, what kind of endpoints we're looking at, or what kind of dose ranging studies that you might be doing, and those kind of things. You want to have a end of phase 1 meeting for that. And so do phase 2 studies. You can do end of phase 2. You can have a meeting there. Pre-phase 3 meeting, you can have end of phase 2 meeting and the pre-phase 3 meeting. It's usually called end of phase 2A meeting or end of phase 2B meeting. Or you can have pre-BLA or pre-NDA meeting. Once you complete phase 1, phase 2, phase 3, when you're submitting the dossier, it's always good to have a meeting there. And at those meetings, we tell you what kind of submission you need. Otherwise, people might give tons and tons and tons of information. Some of them may not even be useful to FDA. So it's good to have a meeting there, right? So this is the process we go through for that development. And type C meeting, there's any other general meeting, anything outside of the development also, you want to go and talk to them. There's type C meeting there. There's a form that you fill it out and for that information. So it's good to know that there is an opportunity to go and talk and see what kind of studies are needed. Now, for the IND, there are different types, as I indicated to you previously in one of the slides. Then so you can have an accelerated development. In the accelerated development, if you have life-threatening illnesses, then you can have so there are two circumstances under which the accelerated development, and one of it, you are using a surrogate endpoint rather than a direct pharmacokinetic endpoint. You can use a surrogate endpoint for that, right? And second thing, when the FDA determines that the safe use of a product depends on restricting its distribution or use. So you need some special population, or you really want to address, for example, I can think of some of those opioid products or some special disease conditions that you use these products where it's restricted from some special population, right? So manufacturers must continue testing after approval to demonstrate that the drug indeed provides therapeutic benefit to the patient, right? The advantage of accelerated development review is the review is done much faster. So these reviews are done a whole lot faster. Now, the treatment INDs, you are looking at any immediate life-threatening disease, means a stage of a disease in which there is a reasonable likelihood that death will occur. Some of the examples of advanced AIDS or herpes simplex, encephalitis, or subarachnoid hemorrhage, some conditions where the treatment IND, essentially, this is often called a single patient IND, where they are made available to patients before general marketing begins, right? So for example, the product is there in a phase 3 studies, the product is not approved, right? But you can give this to a patient, right? Otherwise, the medication may not be available to that patient. So then we will see the exploratory IND. In the exploratory IND, these allows quick human studies. You don't have to wait for all the data, you're just exploring. So it is conducted very early in phase 1, and it involves very limited human exposure. Important thing is it has no therapeutic or diagnostic intent. Sometimes people may question, right, if there's no therapeutic or diagnostic intent, why are we doing, right? So essentially, the product has not been developed yet. The product is actually, see, the guidance does not distinguish between a drug product and a drug substance. So you have a drug substance, which is a chemical, and that chemical gets translated to a drug product, right? So here, so it's not distinguished between a drug substance or a drug product. So it's a very early on study. That's why it's saying that there's no therapeutic diagnostic intent, because the formulation is not developed at all, right? So there are guidances given for exploratory IND. So don't just end up doing a lot of work unnecessarily, because you're allowed to explore very early on. If you want to administer to humans and test, you can do that by exploratory IND. Parallel track IND. Under this policy, patient with AIDS whose conditions prevent them from participating in controlled clinical trials. So you have a patient who doesn't have a medication because it's not approved, and they cannot participate in a trial because because of the inclusion and exclusion criteria. Sometimes they get excluded there. So these are those kind of patients, and they can receive investigational drugs shown in preliminary studies to be promising. So if you have a promising drug and somebody wants to use it, it's not approved, and they're not even a part of the trial, they want to use it. So it does allow, the policies do allow the patients to get that medication. Emergency IND, an emergency situation that does not allow for an IND submission. So even IND is not even submitted. IND is not even submitted, right? So, but there is an emergency situation, right? So FDA allows, this allows FDA to permit treatment of a patient in advance of an IND submission. If something looks promising, there's nothing else, there's no other alternative. It's a serious condition. So you can use emergency IND. So provision is provided. There are provisions for that study. Now, subpart E, there is a section where when there is no satisfactory alternative exists, then you can give that medication. So a lot of opportunities exist for giving drugs to the patients in different forms of INDs. I just thought of highlighting those things. Now, once it goes to the submission after all the IND studies, this is what we need to pay attention as we are developing, as we are discovering and developing the drugs. So it's not like, oh, I knew this mechanism, I knew that mechanism. That's not that important now. Because what's important is we know that what's a medical officer is looking at, what the pharmacology folks are, clinical pharmacology, no, the pharm folks are looking at, what are the chemistry people looking at, what are the biopharmaceutical people looking at. These are drug dissolution and things like that, right? And then what is the statistical folks looking at, what's microbiology. So it has to pass the scrutiny of all these people here, right? So once it passed, then the review is completed. By the way, once the reviews are done, sometimes there is a meeting with the sponsor, or sometimes there's an advisory committee meeting. You may have seen recently for the vaccines, right? Every time the vaccine comes out, whether it's a J&J or Moderna or Pfizer, there are advisory committee meetings to provide recommendation. It's not done all the time, but it's done sometimes, right? The advisory committee opinions are taken, right? So after review is done, you have a sponsor meeting, advisory committee meeting. When the review is completed, if the review is not acceptable, sometimes there are deficiencies, major or minor deficiencies. So you go back and look at it and provide additional information to FDA, right? But if this is completed review and it is acceptable, then you look at the labeling, if the information provided on the label, right? If the labeling information, if it is not good, go back to the sponsor and have them change the label, right? But if the labeling is okay, and then it goes to inspection for the site where it is manufactured. And then if everything looks all right, if the inspection is good, then NDA action either is approved or not approved, right? So NDA action is done. If the inspection site, if there are issues there, there is pending satisfactory results, right? So that's how the NDA gets approved. Just one thing that, a standard review time for an NDA or a BLA is 11 months. But if it is submitted in priority or accelerated development, in the priority development, then it's a 6 months time. The subpart H accelerated approval based on surrogate markers. Subpart I, there's animal rule now. There are drugs that are approved in conditions where human studies cannot be done at all. For like, I remember one drug, Prussian blue, when, if there's a dirty bomb, if your radioactive decontamination is needed, right? How do you do clinical studies for those products, right? So some of those studies are done with the animal rule. So FDA uses regulations and product standards, right, to approve the drug. And then FDA uses the enforcement actions. If something doesn't go well, if something fails, they need to correct all those things. Then they can ask them for a voluntary recall, or there could be injunction or seizures and things like that. And so that will give FDA that. So FDA has an enforcement authority to do that. So some challenges for FDA are the scientific breakthroughs, the pace of the work, especially the pace of the work in NIH and pace of the work in drug discovery is so much, then it takes a long time for FDA to catch up with that development. So it's hard to keep up with rapidly advancing technologies, right? There are more and more sophisticated products. It's unbelievable how sophisticated the products are. And the new public health threats, as you're seeing now, either antibiotic resistant bacteria, and now we are seeing that, the COVID, they have different strains right now. And, we are struggling to develop the vaccine with a certain strain. And now all of a sudden the new strains start coming to understand them, to understand the medications that are effective, and making sure that they're safe and effective, and they're constantly reproducible. What the drug we give today should be same as the drug tomorrow or day after until the expiration date. It should not change, whether it's a drug, protein drug, or a vaccine or a gene therapy product. It should not change, right? So these are challenges. Then international commerce, and then the consumer information. People expect this to know. So all those things, I think, are huge challenges for FDA. So we have seen that. So there is a discovery for the drug, and then there's analytical development, and all those studies are done. And there's a preclinical development. It's followed by clinical development and marketing. Now, so you have a product, right? So either it's oral product or a parenteral product or a lyophilized product. You have a product. The studies do not end here, because after the product gets approved, you might want to make changes there. Either you are changing a site, or you are changing some excipient, or you are changing some manufacturing method or a process. You want to improve the process. So that's why we have to give annual reports for that. And there are different types of supplements depending on the extent of changes. If the changes are not much, if there's an annual report we have to give. If there are some minor changes, then we have to give supplements called CB 0 or 30 or prior approval supplements. Then the post-marketing surveillance is done. Then you do continuous improvements of that product. All that is looked at, right? So there's a tremendous burden on the manufacturer. I wanted to tell you something that for the CMC section, if you're really looking at guidances, then for monoclonal antibodies and therapeutic proteins, you look at CEDA guidances, right? For other proteins, you will look at the CBER guidances. So, for example, you have Office of Blood Research and Review. So you will look at some blood products, right? You will see the reviews there. Office of Cellular Tissues and Gene Therapies, they have provided a guidance for cellular tissues and gene therapies. You can go to this site and get the guidance. Vaccine Research and Review. The reason they have separated guidances are the INDs are highly product dependent. For example, the manufacturing scale. you can sometimes take a single lot of product, you prepared a lot of product, and you can treat thousands of patients with that single lot. Or sometimes just for a single patient, you need multiple lots, right? Some of the phage therapy I've seen, I was involved with one of the work where I was amazed to see how the different phages were obtained, and sometimes the amounts, like for example, a bioreactor, the amounts obtained are so low in a bioreactor, you have to combine several of those lots. And there's a process to do that, to do those initial studies, right? So sometimes you need a lot of lots just for a single patient. So there are different methods, different technologies, different product characterizations, technologies, right? Practicality of testing. It's very difficult to do a comprehensive testing all the times, right? So that's why a lot of things needs to be balanced out to find out the risk versus the benefits to the patient. Now, the risk, so for example, if a product is there in the blood research and review division, right, within the CBER, coagulation factors are derived from either human plasma or culture media from genetically approved engineered cells for replacement therapy to treat patients with congenital deficiencies, right? These are blood products. So it has benefits, right? So we know the benefits of the blood products, right? They're effective in controlling bleeding episodes. Sometimes they could be life-threatening. So these are blood products that are reviewed, right? So what are the potential risks? So when they're looking at INDs and when they're looking at eventual approval, some of the thing, infections due to adventitious agent, right? So the way you are deriving your drug products. So you are looking at, they may be adventitious agents, so there may be infections due to that, or sometimes there's a development of neutralizing antibodies, right? Or sometimes they're just allergic impurities, right? So they will focus on these things. Now, if you are looking at vaccine products, for example, so these are combination vaccine composed of bacterial antigens such as toxoids or polysaccharides. There are different types of vaccine. This is just one example here, right? So benefits, they're highly effective to prevent as well as treat or eliminate disease. A lot of diseases got eliminated completely, right? So they're protecting health of vulnerable population, right? And it can create a herd immunity. A lot of benefits of using vaccine products, right? But there is too. Adverse reactions are not detected early on. The immunogenicity issues, the herd immunity is dependent on immunization rate, and decrease in risk tolerance with declining disease. If a disease is declining, the risk tolerance also decreases, right? So those are some challenges that they're looking at and which the sponsors need to address when they're developing protein products or the vaccine products. Now, cell and gene therapy products, these are autologous, the stem cells selected using monoclonal antibodies, and then they are expanded in a culture, they are matured with cytokines, and then they're giving back to patients. You see, the process is much more involved, right? Unlike the previous ones. So the benefit, they are very potent than current therapies. They're applicable to a wide range of very difficult to treat diseases. Some of the diseases are so rare, right? So you can treat those diseases. They may have fewer side effects. There's a lot of, there's a very targeted approach. So these are benefits, but it has a lot of risk in it, right? Potential for tumorigenicity there in it, cellular contaminants there, the safety of the reagents, since so many of them are involved, the sterility of the product, product tracking and segregation issues, the characterization of these, the insufficient characterization. These are some of the problems of the application that are submitted. Product stability, product variability. So a lot of those things. So when you go to this division of cell and gene therapy products, they are looking at all these things, right? So depending on the office, depending on the product. Now I will talk more about the characterization and more focus on the protein formulation development for a scientist. For example, if this is a protein API, so you are developing, for example, a monoclonal antibody, or deriving it from cells, right? It's taking it out from a bioreactor, so this is just a drug substance. You're not talking about a drug product yet. You're only talking about a drug substance. Just the drug substance, the variability or the problems can come from the way you run the bioreactor. It has a lot of variables, the cell variables itself, the feed variables, right? The processing variables, what's the stirring speed? We have done quite a bit of work with bioreactors, and I'm sorry I did not list publications here. There are a lot of publications, that where we have looked at the variability within the bioreactor. There's tremendous variability that can come from a bioreactor process, and the way they can degrade, and the analytical methods also, right? Either you're using size exclusion, or you're using LC mass spec, or a GCMS, or you're doing bioassays there. So we need to have good validations of these methods there. The environmental conditions, I know one of the monoclonal antibody we were working with, the buffer made such a big difference, right? The activity was so different when, the stability. With one of the buffer, we were struggling that it kept changing. And then when we changed the buffer system, and the protein was fairly stable. So the environmental condition, the temperature, the moisture, the pH, the light, the ionic strength, buffer concentration, the dielectric constant, if it is a liquid formulation, the protein formulations, right? So all these things can make changes. The variability can come from that. A degradation mechanism, you can have some aggregation there, you can have adsorption, you can have deglycosylation. So a lot of reactions can happen there. So that's why it requires a thorough characterization studies for your primary structures, for your secondary structures, for your tertiary structures, right? A lot of those, and the association, quaternary association. We need to have a thorough understanding. This is just for getting the protein molecule as the drug substance itself, right? So some potential steps, for example, if you're obtaining an OPI, right, you have cell banks, then you are thawing. The wild thawing can bring, so we need to have a good understanding and documentation to show that this you are getting consistent results. Inoculation preparation, right, after you freeze thaw, then you have inoculation preparation, then you are running bioreactor. Initially you might be running small bioreactors, then you might be running large bioreactors, right? 100 liters, 500 liters, or even 2000 liters, or even more bioreactors, right? And then you have all this, liquid that's coming out, centrifugation and filtration, downstream processing there. And then you may have to run several chromatographies there, right? For example, ligand chromatography, or you may have to look at metal chelates there. Then you have to do inactivation, the viral inactivation that's done at low pH, right? Then you are doing ion exchange chromatography. Then you are doing vial filtration, and then you are doing ultrafiltration. So you can imagine how many, and then you may have to run some reductive amination or certain reactions there, right? And that involves, after that, it involves even more chromatography and filtration. And then there is a bottling and freezing. And then after all these things, then you are using so many different methods for analytical characterization. So it's a tremendously burdensome process with a lot of potential for variability. So unlike small molecules where there's a lot of freedom for manufacturing, a lot of flexibility, this has very little flexibility, because since the variables are so many, anything can make dramatic changes there in this product. That's why it's a very, very restricted form of development here. So some critical quality attributes for the protein molecules, whether it's a peptide or protein molecules, the appearance is very important. The osmolarity is seen. That's what FDA is looking at, all this information, right? The pH of the product, I indicated to you, the pH could, have a very significant effect on its safety and efficacy and stability. The primary sequence and the peptide mapping for identity, then you're looking at protein characterization. then the product-related impurities, the process-related impurity, the host cell protein, the host cell DNA, etc., and the toxin or virus. So we are looking at process-related impurities, or if there is any glycosylation profile, or there are carbohydrate structure that needs to be characterized. Then you're looking at secondary and higher order structure. You see the amount of work that it takes for the proteins. Now this aspect of the slide, people usually, the scientists do not pay a lot of attention to it, but it's very, very important. If your product really has to see the light at the end of the tunnel. So the previous slide that I showed you was only the drug substance. The drug substance eventually needs to a drug product, because that's what patient is taking, your BLA or an NDA or an ANDA. ANDA is approved new drug application. So, or a new drug application if it's a monoclonal antibody. So if it's a CEDAR application, or it's a BLA or NDA or ANDA. All these products, now these products could be an injectable, or it could be an oral product, or it could be a subcutaneous product, injection or subcutaneous, intramuscular, intravenous. These are injections or solid orals, or different types of dosage forms, right? So previously we have seen drug substance, we have to go to drug product. How do we go there, right? So drug substance need excipients. You have to select the right excipients, characterize it, the product properties, the processing condition, the environmental condition, container closure, scale up, dosage form. All those things needs to be looked at, understood very well to get the final product. Now, one of the things I noticed in some of the academic scientists who are working on, they use certain excipients and then they do a lot of work on it. And the first question when you ask them, why did you use the excipient? They did not have a good answer for it. Oh, we just tried to solubilize it. No, when we're using excipient, there are certain guidelines for using excipient. For example, if the excipients are well known, no questions asked, you don't have to do a lot of studies related to excipient. If the excipients are there in a monograph like USP or some monograph, or FDA has a database, it publishes a list of all the excipients that are used in concentration. It's available free on the website. The excipient you're selecting, just look at the FDA website. It's called the IIG. Just Google it, you will see it. So it shows the excipient has been used. If the excipient is used in a certain concentration for a certain route, and if you are using the same excipient, you don't have to do any extra study, right? If there's a documented human use in the proposed level, then you don't need to do any extra study. But if you have a totally new excipient, or if the excipient is not there in the IIG list, you have a massive amount of work that you need to do. There's a new excipients, there's a battery of tests you need to do. Just to give you a flavor, I have shown some of the inactive ingredients guide, right? IIG lists the amount that is needed. And sorry, I'll in this slide. So the guidance is a May 2005 guidance. It defines what a new excipient means. So if you have a new excipient, chances are if it's not there in the IIG, if it's not there as a GRAS, if it's not there in USP, chances are that it is a new excipient. If there is a new excipient, these are extra studies that people need to do. Normally, they don't do these studies. Nobody wants to do these studies if it's not required, right? So, but if we don't know, you develop something, you progress so much in your studies with an excipient that's not characterized, a firm will have to end up doing all these studies. That's why they don't even want to look at our discoveries, because the moment they see you use some excipient in all your studies, and now they have to justify, they have to do all these studies. So that's why they don't even want to look at those kind of formulations. So you have to do safety pharmacology studies. If the excipient is used for a short term, the disease is for a short term, then the acute toxicity studies, the ADME studies, the genotox studies, 1 month repeat dose studies, reproductive tox studies, all these things need to be done if excipients are used, if the formulation is used for 14 days or fewer. If it is used for 2 weeks to 3 months, intermediate use, all the previous tests, but a 3-month repeat dose study. But if the excipient is used for more than 3 months, then we have to do all the previous tests in addition to that, 6-month repeat dose toxicity, and other tests as recommended, right? And then we have to do a 2-year carcinogenicity test, right? And for non-oral excipient, there are even other requirements. So why do we want to do all that, right? If you use the excipient that's already used by another manufacturer, or if already used in other products, or if it's a grass excipient, you can avoid all these things. So please pay attention when you are doing even your initial studies. Try to work with excipients that are well known. I just wanted to illustrate an example. So you have seen a product, and then a chemical API, when it goes to process, as I indicated to you, there are different processing or excipients or environment or the packaging for the formulation. All those can bring variability. This is just one example. You're lyophilizing something, right? In lyophilization, sorry, it's not 1, 1, 1, it's 1, 2, and 3 steps. You first freeze the liquid preparation that we have. By the way, when I look at lyophilization, one thing comes to my mind, the COVID vaccines that are there, the Pfizer vaccine, it requires deep freezing, right? 70 to 80, minus 70 to minus 80. Then, the other one, the Moderna requires like minus 20, around minus 20. And then, Johnson & Johnson refrigerator. I'm just thinking that, hey, you know what, the CMC, I don't think they had time to spend a lot of information on CMC. They could easily have been stabilized to run for a long period of time, and lyophilization could be a great strategy to improve their stability and increase their ability to store at lower temperatures, and also increase their expiration dates. Right, so in freezing, so in lyophilization, if it's a liquid product, you are freezing, so there's ice crystals, and then your primary drying, so ice crystals are sublimated, right? You sublime and remove the ice crystal, and the secondary drying, where the unfrozen water is removed, right? So this is a lyophilization process. For example, ice nucleation here, minus 20 degrees, you are seeing this kind of ice. If you have minus 15 degrees, you have this kind of ice. And so if you're doing the primary drying, this is just one example I wanted to illustrate because we spent quite a bit of time on this one and published quite a few papers on this one. So if you're doing this drying at this temperature, if this is an uncontrolled, the freezing, this is uncontrolled ice nucleation. You can easily see this is the uncontrolled ice nucleation here. But if you have a controlled ice nucleation, your variability is tremendously reduced, right? This is beautiful, where in uncontrolled one, you have multiple vials in a lyophilizer, so they are freezing at different rates at different times, depending on the location of the vials within the lyophilizer. That's why you're seeing this variability. And this one, it just sublimes very quickly in the controlled environment. So here is a formulation, a monoclonal antibody formulation that we had spent some time on it in-house, just have it in a bioreactor there. And these are some of the excipients we use to make a product to do some of the uncontrolled and controlled experiment. This is an in-house formulation. And the product was evaluated by HPLC, UV-Vis, DSC-TGA, and all those different techniques. The product was evaluated. I'm just trying to show you the advantage of control. That's the experiment we have done to see the benefits. Now, in the uncontrolled, if you see the specific surface area, this one, the control one, is much lower. The recon time, the reconstitution time is lower. The percent moisture, the transition temperatures, the particle size. So this is a better formulation, and also the primary drying time was considerably reduced, right? So it just enhances the efficiency of manufacturing, and also the cake, the lyophilization cake that's obtained, this was much better. That's why the recon time was lower in control. So these are studies people can do to enhance. Then here we are just looking at and control and control some of the CD spectra, showing some data here. So you can get better. I mean, the process can be enhanced, still better. This is one study. So we had increased the process efficiency almost by 19%, improved the quality of the product by controlled nucleation. Like that, studies can be done, right? Now, when we have the products, the expected characteristics for a product, I had shown you before in the previous slide for the drug substance. Now I'm showing you for drug product, right? So appearance and color and clarity, identity, the primary sequence, okay, and the tests that are needed, the glycosylation profile, the carbohydrate structure, the secondary and tertiary structures, the process related, all these things are expected to be submitted, and they will be looked at for the approval of these protein products, right? So all these are expected properties that are needed, right? In addition to that characterization of the product, we also need to demonstrate the stability data, right? Accelerated as well as the real time, right? So there are conditions either there are different conditions for that one, right? So either I think 25 degrees and 40% humidity, or 40 degrees and 25% humidity for certain liquid products. But in the case of protein products, it's refrigerated conditions or very low temperatures, right? So even for stability sample, the appearance, the pH, the protein concentration, the purity with reverse phase HPLC or mass spec, or impurities by size exclusion, or specific biological activity, the endotoxin levels, the sterility, all those things are evaluated for the approval of a protein product. Now, very quickly, the last couple of slides that I wanted to show you was similar to the generic product. For a small molecule, you have a brand product and a generic product, right? So this thing is a biosimilar. It's a definition of a biosimilar or biosimilarity. So if a patent expires for a brand product, now you want to make you don't call it a generic product here, you call it a biosimilar product here. So a new provision came recently from the Affordable Care Act, and that was the original application for a protein formulation is 351A in PHS. The RLD is reference listed drug, the brand product, right? If you want to make a biosimilar, this is 351K, right? So biosimilar or biosimilarity is defined in Section 351 of PHS Act to mean that a biological product is highly similar. This is the requirement. So it's almost impossible to say it is the same as the brand product because it is enormously complex, right? You're talking about 150, 160,000 molecular weight versus a small molecule of 200, 300, 400 molecular weight, right? And then a protein product, you have primary, secondary, tertiary structures, too many things that can change in a protein. That's why it's impossible to say if the source is different, that this product is exactly same as the original product. So the requirement is to show that if they are highly similar, and if there are no clinically meaningful differences, then you can get the product as a biosimilar product approved, right? So if you want to get a biosimilar product, what kind of characterization? There's a lot of physical chemical characterization needed. There's a lot of biological characterization needed. Preclinical work, not much, but it is needed. But a lot of clin pharm work is done. Clinical pharmacology. This is a course in clinical pharmacology, so we need to pay attention that for a biosimilar, a lot of clin pharm studies are done. Of course, then immunogenicity or some other studies are done. Not a whole lot of clinical study after this, but this should suffice for the biosimilar. So to give you an example, if you want to do clin pharm studies to demonstrate biosimilarity, we need to pay attention to study designs, the reference product characterization, the study population, the dose selection, the route of administration, the PK measures. How do you look at the PK measures, right? It's not a small drug that you can just analyze it. It's not very easy. The PD measures, defining the appropriate PD time profiles, statistical comparison of PK and PD results, and if there are any extrapolations among indications, right? All those things are looked at for the biosimilar product. So it's an example here. For example, you have filgrastim, right? So you have so many different, if there's a cancer patient with non-myeloid malignancy, see, this is the dose you're using, IV or subcutaneous. So if you see the label for filgrastim, it shows that this is a patient with severe chronic neutropenia, then this is the dose that is used there, right? If the patient is for bone marrow transplant, right, then 10 microgram per kilogram is used for this one, right? So, so you have different doses that are used. So that's why we need to do some studies, the ClinPharm studies for that one. So when you have recombinant DNA products, when you have monoclonal antibody products, right? So we need to do some ClinPharm studies because the label has different doses there in it. So it's just a small example here. So for a biosimilarity, if we want to obtain a biosimilar for filgrastim, or for example, another, this is a recombinant protein, but you can do it for a monoclonal antibody too. So we have to conduct PK, PD similarity trials, right? Conduct safety trials, right? So PK well characterized in healthy subjects and patients, similar PK after, IV and subcutaneous injection, because that product is used both for IV and subcutaneous, right? Because it's done in different doses, different ways it is used. Normally a product for oral use, you're comparing a small molecule with oral use, right? But here you have intramuscular in some conditions, subcutaneous in some other conditions. So we need to show under those conditions, they're highly similar. So, so you might be looking at some neutropenia, right? And so you're looking at them and then comparing it. You might be looking at some of the biomarkers here. Or you might be looking at some PD endpoint, CD34 mobilization. You can be seeing it. So those trials, you need to show similarity, and then the analytical characterization, we are showing that they are highly similar there, right? So hopefully I provided you, it's just one class. This is an entire course by itself. So I just tried to condense all that information in one presentation. Hopefully I gave you a flavor of the complexities of the protein and things that you need to consider for development, just for both for the API and what kind of characterizations you need. And then from the API, then when you're making a drug product, then what kind of characterizations you need and what kind of stability characterizations you would need and the analytical characterizations you would need. Hopefully I gave you a flavor of those things. And then I also provided some regulatory background so that you can go and look at appropriate division guidance documents. So for example, if you are interested in vaccines, you know where to go and look at the CMC requirement for the vaccines in the division of OVRR in CBER. Same thing for cell and gene therapy. Now you know where to look at those documents there. And also, you know that excipients are important early on to select the right excipient, because if you use right excipient, a lot of studies that you have done may not be of much relevance to the industry. That's why they don't take some of our work very seriously, right? So would like to thank the FDA staff. I've worked in FDA for about 11.5 years as a director and as BRS scientist. That's how I got a lot of this information from FDA. But now I'm in academia, so I still do research, and so I have postdocs and PhD students that are contributing to some of the work we're doing. And some of the work I have done, we have done like product recall. It's called a PQ-FACT team, product quality failure assessment and control team. They looked at a lot of product recalls, including protein product recalls. So we learned a lot about why products fail sometimes, right? So the big team has helped me on that. And then throughout my life, both for the protein as well as the non-protein formulation science and the ClinPharm work, a lot of people have supported the FDA, the NI, the Department of Defense, the Texas State, as well as the university and the pharma and excipient sponsors. So with that, I will stop here. Thank you very much. I'm sorry we are not seeing face to face. Otherwise, we would have had some opportunity to do Q&A, but unfortunately, I have to stop here. I wish you all the best and thank you very much for spending time with me. Thank you.